today Antoine, Gessain, and Manon Curaudeau from the Pasteur Institute. Antoine Gessain is head of the Institute Pasteur Oncogenic Virus Epidemiology and Pathophysiology Unit. He will present the Institute research on Mpox, monkeypox, you know, since this summer we say Mpox, in collaboration with the Muséum National d'Histoire Naturelle, those who have been already since long at SCOR have probably participated to the webinar we organized with the National Museum of Natural History on biodiversity. Manon Curaudeau is a postdoctoral fellow in the unit. She will present the results of the research and scientific advance made possible through the support, the financial support of the SCOR Foundation for Science. Probably for 2 years when we first did a webinar on monkeypox, and we announced that we would support research on monkeypox. Some of you were just slightly surprised, saying, okay, is it really worth to support research on monkeypox? Those persons during this summer were all calling me back saying, Philippe, do you know what is monkeypox? Where can I find something on monkeypox? And of course, I sent them back to a webinar and the research we had been supporting, and also to the Institute Pasteur, and saying, okay, it's not something which is surprising us, and therefore it just underlines how important it is to anticipate with scientific research what may happen in the future. And here, during the summer, most of you who were involved in the questions were able to quickly update their knowledge thanks to this research done by the Institut Pasteur. I will not go further and give immediately the floor to Antoine Gessain. Antoine, you have the floor. Just before beginning, I will mention, please, except if you have a question of understanding, don't ask questions during the presentations, and we will concentrate and gather the question after the presentation. Antoine and Manon, you will have the floor for how much time you need. In any case, we stop at latest at 3:30, 1 hour 50, therefore is the maximum. And of course, I let you make your presentation and take the time you need, between 40 minutes or 1 hour, depending on the message you want to deliver. And thereafter, we will go give the floor to the assistants. Okay, Antoine, you have
the
floor.
Before that, excuse me, may I add a point? Is we will have the third expert speaker today, Alexandre Hassanin, is a lecturer at the Sorbonne University, the researcher at the Muséum National d'Histoire Naturelle. He's specialized in mammalian evolution with taxonomic expertise on bats and criminals, and particular interest in species identified as various reservoir hosts. Alexandre Hassanin is in charge of supporting Manon Curaudeau in her research at the Muséum National d'Histoire Naturelle.
Okay. So thank you very much, Philippe. Thank you very much for the introduction. Do you hear me? It's okay? Everything is working?
Yeah, yes? Good.
Okay. So I will speak about half an hour, and I will give you an overview of what we know about monkeypox today, everything you want to know without daring asking the situation. So as you know, I'm working at the Pasteur Institute. I'm chief of a unit working on oncogenic viruses, but for the last 15 years, we have been working quite a lot of emerging viruses, and especially zoonotic viruses in Africa. So I will speak about 4 chapters. The first one will be a kind of history, virus, genetic variability, and a few words about the reservoir, because Manon is going to speak quite a lot about that. After that, we will describe the clinical and epidemiological aspect of the African classical form. So what is classically monkeypox? And we will discuss after that the 2 different emergents. One is 2022, the different aspect of this pandemic, especially when the WHO Director General declares monkeypox outbreak a public health emergency of international concern. This was 2 years ago, when during the time we were working on that. And about a few months ago, there was a second emergency in 2024 in DRC. So it's the eastern part of central and eastern part of Africa and East Africa, where WHO Director General declares a new pandemic, monkeypox outbreak, a public health emergency of international concern. So what we know about the discovery and the isolation of the virus in 1958. So there was 2 outbreaks of a spontaneous non-fatal pox-like disease in some monkeys, which has been observed in the colony of an institute in Denmark. There was a maculopapular rash. Generally speaking, there was general health with no fatal case in the monkeys. And the virus was isolated from pustular lesion in eggs and in tissue cultures. That is what is usually done for isolating pox viruses. The virus was related to vaccinia virus, which is used in the vaccine against monkeypox. And on electron microscopy, there was a typical brick-shaped appearance of monkeypox, of pox. And because this virus resembled pox, and because it was found in monkeys, the virus was called monkeypox virus. So after that, between 60 to 68, there was other monkeypox outbreak in different colonies of captive monkeys in different places. There was no human cases detected during this outbreak, and this suggests that humans were not susceptible to monkeypox. But it was very probably because all these persons have been vaccinated against variola. The first case of monkeypox in human was reported, as you see here in red, in 1970, during the National Smallpox Surveillance and Eradication Program then underway in Africa. It was a small boy, 9-month-old boy, with a rash on the skin, with different lesions like lymph nodes, and the virus was isolated from his skin lesion. Unfortunately, this poor child die from measles before discharge. Just after that, there were 6 other cases reported, mostly in children, in the 1970 and 71. But it's very important to understand for many, many years, you can see on the map the different cases there at that time, that monkeypox remained exclusively an African disease with only sporadic cases in forested areas of Central and Western Africa, and small outbreak in the human population, mainly in the Zaire at that period, and now it's the Democratic Republic of Congo. Until 2003, where the first cases out of Africa were reported. So these were the first cases of monkeypox out of Africa in 2003. You can see the paper in the New England Journal relating these discoveries. These events occurred in the USA and was linked to the importation of the probable reservoir, we didn't know at that period, of Gambian pouch rats and other animals from Ghana to Texas. And this rodent transmitted the virus to the prairie dog, as you can see here in the center of the slide, housed in the same exotic animal facilities, which then infected humans, mostly young adults and children, because they wanted to have these kind of animals as pets. And the transmission was usually through bites or scratch, and you can see the different kind of primary inoculation lesions on the skin on the right in the picture. Monkeypox was isolated from this animal and from the children affected, and that was the end, I would say, of this small series in the US, about 80 cases, but no deaths, 80 cases in humans. After that, the first export of monkeypox from the African continent by human host, and not, as we have seen just before, by transfer of potential animal infected, was in 2018 with few cases in Singapore, in UK, 3 cases in UK, 1 in Israel and 1 in Singapore, as you can see on the slide. And most of them, all of them, originate from the south part of Nigeria, West Africa. And at that time, there was an outbreak in Nigeria in 2017, 2018, with more than 100 cases confirmed or probable. Important to say that here, most of the people affected were adults. Before that, it was children. There was some deaths, especially in persons infected by HIV. And the distribution and the epidemiological study suggests both zoonosis, that means passage from animal to human, but it was not really well demonstrated. It's why I put plus minus. And mostly were secondary human-to-human transmission, especially in male, young, and HIV positive persons. So one word about the virus. So monkeypox virus belongs to the Poxviridae and the Orthopox genus. It's a very large envelope virus. Each virus encapsulates a core, as you can see on the slide, with a linear double-strand DNA genome. It's a large genome, 200 kilobase, coding approximately 200 proteins. You can see here the typical shape on electron microscopy. And it's very important to understand that these viruses, the orthopox viruses, which contain the variola, the vaccinia, the cowpox, the camelpox, the recently Alaska pox, and the monkeypox, are closely related on the antigenic properties. That means that you can have a cross immunity. And as you can see in yellow, this is very important. The vaccination against smallpox generally protects against monkeypox. And in the classical historical study in the ex-Zaire, you have about 85% of protection. And this phenomena, the diminution especially of the protection, is one of the factors favoring the emergence of monkeypox, as you will see just after. One word about the genetic variability, because we will speak, and also Alexandre and Manon will speak about the different strains, different variants, which are called clade 1 and clade 2. Generally speaking, the clade 1, as you can see here in blue, originate from Central Africa. That was the first described. It's why it's called clade 1. And the second one originate from West Africa, and it's called the clade 2. And it's important to understand that there is only one country known for the moment, Cameroon, just a little between, if I may say, West and Central Africa, where the two clades, the two different strains of viruses has been described, especially clade 1 and clade 2. This is very important to try to understand the emergence of the pandemic that occurs, 2-3 years ago, and also the different kind of reservoir that can be harbored by this virus. So one word about the reservoir. I have only 2 slides because Manon is going to speak really quite a lot about that. Monkeypox is a sylvatic zoonosis, but the reservoir of monkeypox has not really been yet clearly identified. Everybody working on that consider that it is rodents, especially squirrel or rats, living in the rainforest of Central and West Africa. You can see some picture typical of this area. African apes and monkeys are considered mostly as intermediate hosts, but not probably the reservoir. And you can have many different kinds of animals, including rabbits, prairie dog, other rodents, monkeys, which are susceptible to infection in captivity and in the laboratory, but are not considered as the reservoir from which there could be some spillover and emergence. So what are the most likely animal reservoir hosts for monkeypox? So this is a paper done by Manon and the team, and she will discuss quite a lot about that, and you will see that the small arborical squirrel is probably the most likely reservoir of these viruses. So let's speak a little bit about the classical form on a clinical point of view in the African settings, that is before the pandemic and the new emergence about a few months ago. Usually, based on the description classical in the DRC, Nigeria, and in Central African Republic, monkeypox epidemic or small cases occur in small remote villages, usually hard to reach and rural forested areas. Sometimes there is armed conflict, population movement, very poor medical infrastructure, poor patient care and follow-up, and most of the cases are retrospectively studied and often incomplete. You can see the difficulty to do this kind of work in the field. There are different phases, 3 phases on the clinical point of view. Incubation, about 2 weeks, a little less. Prodromes, few days, with fever, headaches, fatigue, and often lymph nodes. You can see here on the clinical point of view, after that, the eruptive phase. So this is the typical clinical aspect of monkeypox on the skin, with macule, papules, vesicle. I will not go in detail. It is usually umbilicated and frequently on the palm and the soles of the feet, which is quite different from the chickenpox. You can have few to several thousand lesions. They are all at the same stage of development, and very important, these are proportional to the density, especially the severity of symptoms. The more you have these kind of things on the skin, usually the worse you get. The evolution is usually into crust with hypo or hyperpigmentation, and in most of the cases, it is a self-limited course, but you can have clinical sequelae, you can have scares, and you can have also complication. It's very important to know that the few people who die really from this disease are due to bacterial skin infection, or it is because it occurs in HIV-infected patients, not really well treated in most of the cases. But I remind you that we are speaking about Central Africa and sometimes in very difficult to reach areas. The case fatal, so you can see the most frequent clinical signs, the rash, the fever, the headaches, pruritus, lymphadenopathy, classical. And important to understand, I just told you that before, that the case fatality rate in the classical setting, people considered that it is lower regarding the West African strains than in those infected by the Central African strain. It is not so easy. I really consider that it depends on the medical situation where you have been diagnosed and if there is some good treatment. Just to give you an idea, it is about 6%, the case fatality rate, the lethality in Nigeria, but can reach 10 to 15% in Central African Republic. As I told you before, the poor person who die usually are children with bacterial skin infection and sepsis, and also HIV positive person without antiretroviral therapy. So what was the typical epidemiological aspect in the African setting? There is a large increase in the number of cases over the last 3 decades, especially in ex-Zaire. You can see here in blue, light blue in the middle of the map, with quite a lot of numbers. That was before the last few years. There is an increase also in the median age of diagnosis, and usually this is due to the large majority of no vaccination against smallpox. How is done the transmission? How you can get infected? Either it is from animal to person, what we call zoonotic transmission, the first one, via lesion or biological fluids from an infected animals, probably mainly rodents. But it's during hunting, butchering, or eating game. It's not because you eat game, it's because you manipulate game and you are in contact with your skin, with the skin or biological fluid of animals. And also it can be human-to-human transmission. And in most of the cases, it's through biological fluids and infected skin lesion of patients. But in many cases, in the epidemiological studies in Africa, the exact origin of transmission is often unknown. Just to give you some example of transmission, especially work which has been done under the responsibility of Emmanuel Nakoné from the Central African Republic, Arnaud Fontanet, Jean-Claude Maniguera, and other people from the Pasteur Institute, especially Camille Besson, who has done her PhD the last few years. You can have nosocomial transmission. You can have intrafamilial transmission. At that time, which is very different to just what I'm going to say just before, after, the sexual transmission was very poorly documented. The transmission chains are generally short, and the classical reproductive number, what we call the R0, which has been well known for the COVID pandemic, is estimated to be around 0.6 to 1. This is quite low as compared to many other diseases where the transmission is much higher. So this is the first great part regarding the form in Africa. That is before the pandemic in 2020 and the new one in 2024. So what's happened now in May 2022? A series of monkeypox cases were identified in the UK, in Portugal, in Italy, primarily in men who have sex with men. You can see the different papers and the different time of and period of the diagnosis. And the health authority rapidly established at that time that we were at the beginning, this was exactly 2 years ago, 2 years and a half ago, of an outbreak of a new clinical and epidemiological form of monkeypox, which was quite different to that observed in Africa and that I just described to you. So the first case in 2022 in the outbreak was confirmed in the UK on 6 May, in a man traveling from Nigeria. But at that time, there was already some cases which were not related to travel to Nigeria. And that means that you had earlier onset date of symptom, probably in late April 2022, as you see on the scheme. So at that time, on July 22 of 2022, WHO declared global health emergency. During the next few months, there was nearly now 100,000, but at that time there was about 80,000 cases of monkeypox, which has been reported in more than 100 locations worldwide, and in countries which were not at all historically pandemic. It was a real pandemic, which means an epidemic in the different continents over the world. It was mainly in US, quite a lot in Spain, in Brazil, in France, and in many other places. Most of the cases occurred in men who had sex with men. So really, that time it was very different to the clinical and epidemiological situation in Africa. The most of the transmission occurred through sexual contact. Just to give you an idea, that was one of the first large papers published in the New England Journal of Medicine, you know, the best journal in the world for clinical studies. As you can see on the red arrows, about 38 years, the median age. Most of them were male, 99%, homosexual men, mostly white at the beginning, HIV positive in quite a larger proportion, and from Europe. The situation changed, as I told you, because after that you have a lot in US, a lot in Brazil, quite a lot in other different places in the world. Just to give you an idea about the evolution of this outbreak, of this pandemic, you can see on the left the situation about 2 years ago. You can see that most of the cases occurred in the US and also in Brazil, in yellow and red. You can see the evolution on the right with the peak occurring for the world around August 2022, and after that slowly decreasing, like the typical epidemic in the world, in the United States, in Spain, and different countries. Below you can see the situation in Europe at that time, the situation in the middle in France. Most of the cases were focused, especially, you know, there was something like 2,000 or 3,000 cases at that time in Ile-de-France, the center of France. And you can see also the histogram with the evolution and the peak in on week 26, and after that the decrease given the numbers given by Santé Publique France. So what was the clinical aspect of this disease as compared to the situation in Africa? It was the same kind of disease, but it was a kind of new pattern. The incubation was a little lower. You have quite a lot of things on genitalia, anal and perianal areas like rectitis and oral lesion with pharyngitis, and these were published in the New England Journal of Medicine. And if you compare very briefly the two different forms, on the left, the classical form, on the right, the new clinical epidemiological form. I will not go in detail because it's more clinical and medical features, but you can see that regarding the location, the affected population, the epidemiological features, the transmission, dissemination, the clinical phase, the symptom, and the case fatality rate, it was quite different, despite it was the same kind of virus, of course. So in this scheme we published in the New England, this is a kind of natural history of monkeypox at that time. First, on the left, you have the rainforest with a reservoir host, some kind of rat, some kind of squirrel, very probably. Below, you have the exotic pet trait that I told you just before with the zoonotic transmission occurring in children in 2003 in the US. But in most of the cases, the situation even now, you have zoonotic transmission in red, as you can see, from the reservoir host to children or hunter, for example, in Central African village. After that, you can have small sporadic or small epidemic locally with few cases in the family members, usually human-to-human transmission. And during the large pandemic in 2024, you have an out of Africa spread of these viruses originating from Nigeria, as I told you, with something like 100,000 of persons affected over the world, mainly men at that time. So this is the current situation in 2024 regarding this pandemic. You can see that there are very few cases now. Just to give you an idea, there is something like 20, 30 cases a week, a month only in France. So it's not nothing, but it's not a lot. And you can see on the left the confirmed case and also same kind of things regarding on the right. So why it disappeared, if I may say, why there was such a decrease, it was due to information and prevention effort and the use not only really on therapeutics, because I will tell you just after, there is no really good therapeutic regarding monkeypox, but vaccination works quite well, especially when you focus on population at risk, mainly gay, bisexual, and MSN, and other partners. So the epidemic of 2022 was remarkably controlled by the mobilization of targeted high-risk population. There was a lot of behavioral modification to avoid monkeypox, and also the high uptake and effectiveness of anti-smallpox vaccination, especially the modified Vaccina Ankara vaccine developed by Bavarian Nordic, and this is still the vaccination that you can use today. The use of treatment like tecovirimat and brondosilfovir, now at the beginning people were thinking that that was working a little bit, but now it has been demonstrated, at least for the first one, tecovirimat, that the effect is not really great. So unfortunately there were some severe forms, and especially during the pandemic, you have nearly 100,000 cases. There was around 200 deaths, and most of the deaths occurred in our regions, especially in HIV-infected patients with really advanced HIV-infected, as you can see here, but also in Africa, especially in Nigeria. That was the same kind of things, and the few children who die, unfortunately, it was irrespective of HIV status, and it was because of the lack of use of rehydration, antibiotic, and so on. So this virus, which occurred during the pandemic, was slightly different to the classical type that we found in Africa at that time. It was called clade 2B, originating from the West African clade, the clade 2, and it began in 2015, 16, 17 in Nigeria before the migration of the infected population. Just to give you an idea, this is a little bit technical. That was a paper in Science demonstrating that the clade 2B on the top, on the left, have some mutation which are specific of the action of an enzyme called ApoBec, and it is only because it is an adaptation of the human-to-human transmission. Because it's not usually, you know, this virus does not grow in human, if I may say. It is not transmitted from human to human quite a lot, but from animal to human at the origin. But if there is a sustained intrahuman transmission, you have the use of and the effect of this enzyme which modify some structure on the nucleotide, and you can clearly differentiate that when you do sequencing. Very interesting, very recent papers done by a large group of persons from Nigeria, from Cameroon, including Centre Pasteur du Cameroon, from the US, from the MIT, from the Script, from La Jolla, and from also English researcher, demonstrate clearly that the southern Nigeria was the origin of the human epidemic I just told before, the pandemic. But at that time, they demonstrate also, this is very important, that there was still some ongoing zoonotic transmission in Cameroon and Nigeria, underscoring, of course, the continuous risk of monkeypox, which means the possible re-emergence of such a virus. And that's exactly what's happened just now, and I will tell you that in the next slide. What is now the situation? And we enter now a new outbreak, very different to the situation that I just told you before in the homosexual population. This is completely different and a new outbreak occurring and beginning at the beginning of this year. So since December 2022, there was an increase of cases of monkeypox in the ex-Zaire, in DRC. Just to give you an idea, in 2023, there was about 15,000 cases. This can be suspected of probable or confirmed cases, but with at least 654 deaths. And at the beginning of 2024, there was something like 4,000 cases, the beginning of this year, with 270 deaths. And at that time, as you can see on the scheme on the left, you know, it is the black dot, and the black is 2024. You can see the increase during the first weeks regarding the number of cases and the number of deaths. And this was what we had, you know, at the beginning of the year. Now, at the beginning of 2023, but published at the middle of the year, especially in June or July, there is another kind of epidemic, a real epidemic, in the eastern part of DRC. This is a purple large dot that you can see on the map in the Kamitunga region near Bukavu city, and also in the eastern African country, quite related, neighboring Burundi and Rwanda. And this is a densely populated, poor mining area. And as you can see on the histogram regarding male and female on below on the slide, you can see there is quite a lot of male, but also quite a lot of female. Usually females are younger, and you know, the people working in the mine have a sexual relationship, heterosexual relationship with a woman living in this area. It could be young girls, if I may say, or sex professional, frequent sex worker. And this occurred in this context of an epidemic of intrahuman transmission. Just to give you an idea, exactly like what we just described before regarding the emergence of a new clade during the pandemic. It was called the clade 2B at that time, as I told you. Now there is an emergence of a clade 1 because it is in Central Africa. It's called the clade 1B, and it is the same kind of modification on a genetic point of view with the action of this enzyme ApoBec, which modify genetically a little bit the viruses. And you can clearly see that the situation now in the eastern part of the ex-Zaire in DRC mirrors by a certain aspect the 2017 and 2018 outbreak of clade 2 in Nigeria. So it's important that to understand that you got something in 2022 and something new now for the last few months in East Africa. Just to finish, as you will see, WHO declared global health emergency on August 24 this year, about 3 months ago, 3 months ago. And on this different curve, you can see, for example, on the left, in DRC, from around 12,000 cases, now it reached, and that was on October 13, more than 40,000 cases. In Congo, there is a slight increase also, despite the fact the numbers are lower. In Cameroon, it is the same. But more important, there is a dissemination in the neighboring countries of the eastern part of DRC. And just to give you an idea, in Burundi, where there was not a single cases at the beginning of 2024, you have now more than 1,500 and now more than 2,000 and even more in Burundi. And this is the clade 1B, and this is mostly heterosexual transmission. But as you get virus from man to woman, when you have virus in a woman, you have now also dissemination a little bit in the family. So in this epidemic, it is not only focused on men and women, but also in children. Just one word before the conclusion. There is other kind of viruses very related to that in other places of the world. And there have been some cases called Alaska pox occurring in the US, in Alaska. And just to give you, in 2024, so a few months ago, that was the first report of the very severe cases of Alaska pox, and which led to death in a person with an history of drug immunosuppression, secondary to cancer treatment. And very probably it was a contact with a stray cat, and which probably transmits the virus on a zoonotic point of view from the cat to the guy who died after that. There was some studies which has been done by the Center for Disease Control in US, and they found that small mammals were probably infected, and especially what we call in English redback voles, I don't know, or voles. And this is a kind of campagnol at dos rouges. This is a kind of rodent that I don't know very well, but just to tell you that there is quite a lot of other kind of viruses related to monkeypox. So just in conclusion, I would say that the 2022 and 2024 monkeypox outbreak remind us something which is extremely important, is that viral emergence is a permanent phenomena without boundaries and often unpredictable in its nature, its target and magnitude. And very probably it's a never-ending story. So to acknowledge the different people who participate to this work and help me to do this kind of presentation. First, you have from the Institute Pasteur. I don't know what's going on.
It's good.
So,
we
Emmanuel Nakoné from the Institut Pasteur de Bangui and all the team. Alexandre Hassanin and Manon, of course, and you are going to listen to both of them in a few minutes. My team at the Institut Pasteur in Paris, especially with Nicolas, Manon, and Olivier. Jean-Claude Manuguera, also at the Institut Pasteur in Paris. And the team of Arnaud Fontanet, especially with Camille Besson, Tamara, and Laura, we have done quite a lot of work in the field. And the ANRS also with Yazdan Pagna, who helped me quite a lot to write the review in the New England Journal of Medicine. So thank you very much. I hope I have not been too long and a little clear, I hope. And now I will give the floor to Manon.
Yes.
Or if
Thank
you have
you
a
very
question,
much.
it's up to you now or after.
Maybe I suggest, Antoine, we do first the presentation of Manon, and thereafter, and then the presentation of Alexander, the side, and then we gather the question. Please, Manon, you have the floor.
Thank you very much.
Allez. Voilà. Tu modifies là si tu veux.
Yeah. So thank you very much. So I am actually a PhD student and I'm finishing my PhD in a month. And then I will be a postdoc researcher, hopefully. And I'm going to present what I did during my years as a PhD on the animal reservoir of monkeypox. And my PhD supervisor were Antoine and Alexandre that you're going to listen soon. Just to remember, but closely, very rapidly, because Antoine just discussed it, but Mpox is caused by a virus, the monkeypox virus, that is a poxviridae, exactly like smallpox, and especially those two viruses are orthopox viruses with double-stranded DNA genome, and they're quite a big genome of about 197 kb. Briefly, monkeypox was first discovered in monkeys in a lab in Copenhagen working on polio viruses. And then it was also found in many labs in the US, Netherlands, and France. And in 1970, it was first found in a small boy in the Democratic Republic of Congo. And following that, there were several cases that were described in Africa up until the 90s. And in 1996 and 1997, there was a huge outbreak in DRC that had many more cases than was already described before. And following this, a lot of cases were described every year until 2021. And so we think that most of these cases are due to the end of vaccination against smallpox that happened in 1984. And because the vaccination against smallpox also protected against monkeypox. And there is also a probability that some of the cases of smallpox described in Africa were actually cases of monkeypox that were not known at the time. There were, as Antoine told you, several exports out of Africa in 2003 and also starting from 2018. And just as Antoine told you, there was a pandemic in May 2022 that affected the whole world, and the onset of the epidemic were at different times in Europe than North America and then South America. And you can see that even in South America, it was not at the same times exactly. And currently, there is a new major outbreak in the east of the Democratic Republic of Congo that is mostly clade 1b, whereas in 2022 it was clade 2b, but I'm not going to focus on that. So, as it was said before, monkeypox is a zoonotic disease, but the reservoir is still unknown. So my PhD was to use a multidisciplinary approach to identify the animal reservoirs of monkeypox virus in Africa. So a reservoir is a species where a virus lives, reproduces, and is sustained. And then it could be like a virus can be transmitted to humans, and that's why we call it zoonotic. And first, there are cases where they are in contact with the animal reservoir. They are the index cases. And when they're transmitted from human to human, we call them secondary cases. But the cycle of the virus could be a bit more complicated with intermediary hosts. And in the case of monkeypox, we think that intermediary hosts could be primates. And even more complicated, it can be not just one reservoir species, but many reservoir species. So this is the idea that we have about this virus. Our objectives are to enhance knowledge on the biodiversity of the most probable reservoirs of monkeypox, but also to supply public health authorities with key data on these reservoirs to aid in the monkeypox prevention. The goal is to support this prevention, to reduce the risk of spillovers from the reservoirs to humans, and then thus lowering the chances of monkeypox outbreaks. So I started my PhD by doing a bibliographic study to identify potential reservoirs or secondary hosts of monkeypox, because we know that this virus belongs to the genus Orthopox virus, which is a genus that is exclusive to mammals. We focused on mammals, but as you can see, there's almost 6,000 species of mammals, so it was complicated to study them all. So we also know that monkeypox human cases originates from sub-Saharan Africa. So we thought that monkeypox reservoir is a mammal that is living in sub-Saharan Africa. And also all African human cases are located in tropical forests. So we think that monkeypox reservoir is a forest-dwelling mammal. But this leaves us with 928 species, which is a bit too much to study. So we decided to focus on the 19 genera of mammals which have at least one evidence of presence of monkeypox in them. So this kind of evidence are virus isolation, monkeypox fragments that were like DNA fragments that were amplified by PCR, or the presence of anti-orthopox virus antibodies. But as Antoine told you, these are not super specific to monkeypox. They are shared by many orthopox viruses. So this is the least how to say that? This is the not least specific evidence. So because there's a lot of taxonomic problems in rodents in Africa, we decided to not study only the species that were discussed as potential reservoirs, but all the species that belong to the genera of the species. So it left us with 213 species. You can see here some of these species. We had a strategy to identify the reservoir in 3 different steps. We, during my PhD, did mostly the two first one, and I began to do the third one. So the first thing was to do ecological niche overlap analysis between monkeypox virus and African mammals, and this was published in a paper in 2023. Then I focused on phylogenetic and phylogeographic comparison between monkeypox and its reservoir. And I started to implement a method to detect monkeypox virus in museum specimens in these reservoirs. So I'm going to first talk about the ecological niche. There was already some study that was done on ecological niche on monkeypox in the same team in 2007 and 2012. These 2 papers used occurrence cases that you can see here and on both maps, but there was a mix between index and secondary cases, also a mix between confirmed and suspect cases, and a lot of suspect cases are actually chickenpox because the symptoms for both infections look quite alike. And also the GPS points that are used are sometimes the GPS points of the villages of the person, which is quite close to which we think are quite close to where they were infected. But sometimes it is also the hospital they were treated in or the health center. So this is an administrative region, so this is not very clear if it is close by where they were actually infected. So also, this is done in 2007 and 2012, so we needed a more updated niche of monkeypox, and we tried to do it with only confirmed index cases. For that, I looked at the literature, and for each described cases, I went back to Google Maps or OpenStreetMap to try to find the location of the villages. And when I was not able to find them, I've looked at US Army maps that you can see here, and I was able to find some of the villages, for instance, the village of Ebara and the Democratic Republic of Congo. And then I put them back on Google Maps to have the same geographic reference. I was able to find 109 reliable GPS points, and when I filter them so that we don't have points that are too close one another, I had 96 that you can see here. I correlated these points with bioclimatic and topological variables to reconstruct the monkeypox niche. So you can see that these variables are based on precipitation or on temperature, So it can be annual mean temperature or annual precipitation, but also on solar variation. And I took the least correlated ones to do my ecological niche analysis. So with that, I was able to reconstruct a monkeypox virus niche that you can see here on the left. And when we compare it with African rainforests, we can see that we have three main groups. One that corresponds more to the Congo Basin forest, one that corresponds more to the Lower Guinean forest, and one that corresponds more to the Upper Guinean forest. And these different groups are separated by either the Dahomey Gap or the Volta River for the two Guinean forest blocks. And between the Lower Guinean forest and the Congo Basin block, we have probably the Sanaga or the Cameroon volcanic line that is acting as a barrier. Overall, our niche is quite similar to the ones that were published before. But there are key difference, mostly that there was this separation between the groups that is not very well seen in the two niche, but also in Gabon, we have a gap in our niche, but the Gabon point is not the one that we can trust the most, because we don't have the specific location of the village, and also there was no cases in Gabon since the one that were described, and this is a country where there is a lot of prevention and a lot of surveillance, so we think that it could have been imported before. Then I used, for animals, I used a different database that are specimen databases. So it's databases that are recording the location of the specimens that are in natural history museums or scientific institutions, but also an observation database that is curated by researchers. And I also correlated that to the distribution database. So that I took only specimens that are supposedly in the known distribution. Then I had, with this result, I had 99 species where I had enough occurrence data points to reconstruct a niche. And we are thinking that the others are species that are probably too rare to be the reservoir because they're very rare and probably not in contact with humans. So I reconstructed the niche of the animals, and then I compared them to monkeypox, and I ranked them. The one that has the most closely, let me rephrase that. The one that is ranking the highest when we compare the niche between the two niche is a small arboreal squirrel that is called Phoenicurus aniratus, that you can see here on the right. You can see that we have also a block in Congo Basin and in the Upper Guinean forest, and also Lower Guinean forest, and also the Upper Guinean forest. And that the area with the highest intensity are the same proximity between the virus and the squirrel. In red, you can see the supposedly the distribution of the squirrel, but you can see on in West Africa that we're all supposed to find this animal, but it is not known from this place. So it might be something to look at. The second one by ranking is Grapheus laurinus, which is also an arboreal rodent that is closely related to squirrels. And then the third one is another squirrel of the Phoenicurus genus. You can see that the niche is quite close, but it is there is no separation at all. So the more we go down the ranks, the least closely related they are to monkeypox. And we can see that the fourth one is also an arboreal squirrel of a different genus this time, Elioscurus reprobrachia. And after that, they're not even really comparable between animal and virus. So this was published in 2023 in Viruses. And the conclusion of the article is that monkeypox niche is fragmented between at least 3 African rainforests, Congo Basin, Upper and Lower Guinean forests, that the 4 species that show the best niche overlap with monkeypox are all arboreal rodents and arboreal scuromorphs. And then that Phoenicurus anerythrus is the best candidate for the reservoir. We have the best overlap metrics. Both of them are the highest ranked. Also that the areas of higher probabilities of occurrence are correspond to the one of monkeypox. And based on what was known before, it is an animal where we isolated the virus, where there is a PCR that amplified some fragments of the DNA of the virus, but also there were like some antibodies found in these squirrels. Just to talk again about the fact that we have a probable presence of Phoenicurus anerythrus in West Africa. You can see here that both these squirrels look kind of similar. And in fact, this one, the one on the left, Phoenicurus anerythrus, was described first as a subspecies of the second one on the right. So it is possible to think that some of the squirrels from the right, some of the Phoenicurus pyropus, in West Africa might be Phoenicurus anerythrus. So we would have Phoenicurus anerythrus in West Africa, and the other would be Phoenicurus pyropus. So this needs to be investigated genetically, so looking at the DNA of these two animals. So one of the other conclusions of our paper was that maybe some pyropus were actually Phoenicurus anerythrus. So what I did then was to do some comparison of phylogeographic patterns between monkeypox and Phoenicurus anerythrus. So we imagine that if we have a perfect coevolution between the virus and its reservoir, we would find exactly the same patterns in both of their phylogenies. But if there was no coevolution, we'd have different patterns of evolution of the two. So for that, we will do some comparison of phylogeographic patterns and some datations. I don't know why the head went back. Sorry about that. So what is known about the phylogeography of monkeypox is that there are like two main groups, one in West Africa and the other in Central Africa, and with smaller groups within the two clades. So the one A and so sorry, 2A and 2B, and in Central Africa there are like many different groups. And here there's only clade 1A. It is a paper we published in 2021. So 1B was not known at the time. And we think that the deep divergence between West Africa and Central Africa might be linked to the reservoir isolation. And we would find some, if we were to find some coevolution between monkeypox and its reservoir, we would have the same groups between the virus and its reservoir. So for that, we collected some specimens in different museums around the world. So we were sent by these institutions fresh specimens, so biopsies of organs that are kept in alcohol. They date back to maximum 30 years ago. But also, Alexandre and me, we also sampled skins, so animals kept as skins, as you can see here, of more ancient DNA, so it's specimens dating to the 19th and 20th century from the Muséum National d'Histoire Naturelle, but also two other museums, including one in Belgium. And you can see that all the squirrels that we gathered are located in Africa, and the only they are like encompassing almost the whole Africa. We're just missing some data from Nigeria. So for the fresher specimens, I did molecular analysis. So I did some amplification of the cytochrome C oxidase 1. That is the fragment we usually use to identify specimens, especially animals. And here you can see the tree that was made out of these sequences. You can see in red that there are like a lot of misidentification with specimens that are not assigned to the right genera. And in gray species, there are like specimens that are in the right genus, but are not assigned to the right species. So there's many identification errors. And we also if we look more closely, we have the Phoenicurus piropus from Central Africa that are here, whereas the Phoenicurus piropus from West Africa are here. So this group is not monophyletic, and especially the one from West Africa are closely related to Phoenicurus aniretrus in Central Africa. So this is confirming the niche result that some related squirrels of Phoenicurus aniretrus are present in West Africa, and we can actually say that all of these are Phoenicurus aniretrus, whereas Piropus is only present in Central Africa and is closely related species, but not in the same. If I redo everything taxonomically, we can have here the phylogeny of Phoenicurus. And when we're looking closely at the phylogeny of Phoenicurus aniretrus, we can see that it has a phylogeny that is similar to the one of monkeypox, with two main groups, one in West Africa here, both of them there, and another one in Central Africa. And each group we have subgroup. So we have a group in an orange that corresponds to West Africa, but on the east of the Volta River or Dahomey Gap, and on the red group that is west of this river. And in Central Africa, we have, I colored it in green, but we have a group that is in the Congo Basin and one group in blue that corresponds more to Cameroon. But this analysis is based on a small fragment that is represent only the maternal lineage. So we need to do some genome sequences to validate these results. So that's what we did. We sequenced the complete genome of 32 Protoxerani squirrels. So we have 10 Phoenicurus aniretrus, 6 from West Africa, and 3 from the Congo Basin, and 1 in Cameroon. And for outgroups, we also sequenced other Protoxerani squirrels, including 12 Phoenicurus, including 3 Piropus that you can see in purple here, 1 Protoxerus, 5 Paraxerus, and 4 Eleuthurus. And you remember that Eleuthurus were also quite highly ranked in our niche study. We pre-processed the data, and we were sadly we had to remove one of the specimens we sequenced. Then we did some selection of genomic markers for the phylogeny. So we used the complete mitochondrial DNA for the mitochondrial data sets. It is about 16 to 70,000 base pair. And we assembled the genome de novo and annotated with mito-Z. And for the nuclear data set, we used some exons of genes that are present in only one copy in the genome. So we had a data set of about 1,017 exons, and they are both present in squirrels and humans with a GC content about 40 to 60%. And these exons are more than 1,000 base pair in Marmota, which is the alpine marmot, but is a squirrel. And then we did the mapping with next-gen map, and then de novo assembly of the reads that maps. For this exon, we decided to use to make a new reference because the Marmota is not super close to our squirrels, and so we mapped the reads of our 10 Phoenicurus aniretrus on these 1,000 exons. And we used the ones that are completely present in at least one Phoenicurus. And we made a chimera reference with all of these. So we can see that almost like that all the specimens did actually contribute to this chimera reference. And then we mapped, again, all the reads to this chimera reference. And we use only the exons with no missing data. And it is 167 exons that we have. And so based on the chromosome, you can see that there are actually we have almost all the chromosomes of squirrels that are represented in the data set here on the right. So then we did some maximum likelihood trees with IQ32. for both the concatenation of the 167 exons, but also each gene individually, each exon individually, and also on the right, the complete mitochondrial genome. You can see that both trees are actually quite similar, and that the only differences are within groups or for groups that are not very well supported. We have the same groups for Phoenicius and Erythrus, with one group in West Africa and one group in Central Africa. And we need, within these groups, we still have like the one that is on the west of the Volta River and Dahomey Gap, and the one on the east, and the one in Cameroon, whereas the others are in Central Africa. So we can see that we actually do have the same patterns as for the viruses, monkeypox viruses. And this is a part we need to work a bit more on, and Alexandre is going to talk about it. So when we're looking at datations of these sequences, we have a difference between the squirrels and the virus. Here the dates are in million years, whereas the dates are in years, and so we need to do more work to try to reconcile the dates in both, because we think that the dates in the virus are maybe erroneous. So we're finishing the paper. We're gonna soon publish it. But the conclusions are that Phoenicius and Erythrus is present in both Central and West Africa. That the mitochondrial tree and the nuclear tree are similar. So for further studies, we can only use the mitochondrial genome. So it is quite reliable. And that Phoenicius and Erythrus has the same phylogeny as monkeypox virus, with the same dichotomy between West and Central Africa, and that the stew clades are separated probably by the Volta River, and that we also have several clades in Central Africa, and we need to identify exactly which one it corresponds to in the virus. When we are using conventional substitution rates for datation study, the divergence dates of monkeypox and Phoenicius and Erythrus do not match, so we need to do more investigation on monkeypox substitution rates. And in perspective, what we'll probably do in the future with another PhD student that will begin soon is the detection of monkeypox virus in museum specimens of Phoenicius and Erythrus. So it is a museomic study. Monkeypox virus has a double-stranded DNA genome, as I told you in the beginning, and DNA molecules are much more stable than RNA molecule. So it is possible that monkeypox virus could be preserved. Actually, its DNA could be preserved in museum specimens. So as it's the case, there was a study in 2018 by an American team that found monkeypox DNA in Phoenicius specimens, but it is a small fragment that they amplified, and specimens from the Royal Museum of Central Africa in Belgium and the American Museum of Natural History in New York. They found positive specimens in 9% of their specimens, so 93 specimens, and in 5 species, including 2 species that were not found as potential reservoir of monkeypox before. However, there is very low variability in the small fragments with no phylogenetic signal and only positive specimens from Central Africa, as you can see here on the maps. So we want to replicate this study, and to study if monkeypox DNA fragments are present in the skin of squirrels. We extracted DNA from 303 specimens from the late 19th and the 20th century from the Muséum National d'Histoire Naturelle in Paris. What is going on?
Hola.
Sorry. I'll be back to the presentation.
Oh.
Here. Sorry. So we extracted DNA from these specimens from the Muséum National d'Histoire Naturelle and the Royal Museum of Central Africa. That's the museum that was in the study before. And we amplified first the cytochrome B to check if samples have DNA, and we have 131 specimens that were where we could amplify DNA. Just a note that all Phoenicius pyropus from West Africa that we have in this sampling are actually Phoenicurus anilatrus. And then what we want to do is to amplify short, around 100 base pair variable fragments of monkeypox to have phylogenetic signal. And because we want to do several fragments, we will be able to cover different regions of monkeypox genome. But because monkeypox genome is what we call a moat, we could not do it before studying the squirrels. That's why we are going to do this after that. If we found some DNA of this virus in collection specimens, we will first sequence the mitogenome of the squirrel for phylogenetic studies, and then we will sequence the monkeypox genome. This will help in molecular datation and serve as a better estimation of monkeypox substitution rates, and probably it will help us to correlate it with squirrel divergence states. So in conclusion, both the niche analysis and the phylogenetic patterns suggest that Phoenicurus anilatrus is the main reservoir of monkeypox, and that is the one that we need to study more. Thank you very much.
Thank you, Gessain. Yes, maybe I suggest we don't stop immediately and we give the floor to Alexander.
Yeah, so I stopped the sharing and I leave Alexander to do it.
So I try to share my screen.
Yes. And I apologize, Alexander, because I forgot to mention your name at the beginning, having prepared my introduction a little too soon before your participation was confirmed, and therefore I apologize.
I'm looking, okay, maybe. Do you see my screen or not?
Yes.
Yes.
Okay. I have just. It's okay.
Faut le mettre plus grand. Mets-le plus grand si tu peux.
Ah bah c'est.
Plein écran.
Je suis au maximum. Alors je sais pas ce qui se passe.
Alexandre, si tu peux faire un diaporama.
C'est bon?
Tu peux pas mettre diaporama?
Je suis en diaporama. je sais pas ce qui se passe.
C'est pas grave, on va essayer, on va voir.
I will put it.
Là, ça a disparu, là, on voit plus rien, sauf toi.
Okay.
Je vois votre slide. Je crois qu'il vaut mieux revenir à ce qui était avant parce qu'il était plus gros. Et puis, si des fois, on n'arrive pas. Voilà.
Okay.
Non, cette fois-ci, il a disparu totalement.
I try again.
Non, même si demain en plus petit, de toute façon, il y a trois diapos.
OK.
C'est OK?
C'est bien?
Ouais,
okay,
on voit.
Fine.
On voit. C'est pas énorme, mais on voit. Vas-y.
Actually, I have just 3 slides, so it's not so long. And I would like to try to explain why do we need to sequence monkeypox virus genomes from squirrels, actually. Oops, of course. So I'm not able to change the slide. It's okay.
The slide disappeared.
Non, il n'y a plus rien.
Votre slide.
Maybe I will try from my side.
Maybe yes, Zing, you could do that. And then,
sorry.
Have you seen that?
Alexander, then you ask Zing to change the slide when needed.
OK.
Là, on voit.
Demandez à Zing de changer le slide quand c'est nécessaire. D'accord?
Okay. But I have to see the screen with the slide. So
you don't see
it.
It's the first slide.
Okay.
Could you la voir?
Maybe, I don't know. Is it okay? Okay. Yeah, you can change the slide, please.
Okay.
Yeah.
No. Oh, okay.
Voilà.
This one?
Yeah. Just one time, if it's possible.
Okay.
So first of all, it's important to note that the monkeypox virus genomes are evolved with a wide variation in substitution rates between lineages. And to illustrate this point, you see here a maximum likelihood tree recently published by Vakaniaki and collaborators. And to illustrate the variation in substitution rates, you see that the longer branch here indicates increased in rates of substitution. So there are three human clades concerned by an acceleration in substitution rates. So first of all, and this is well known for a long time, in West Africa, we know that human strains have evolved approximately 5 times faster than those of the clade 1, so the clade only found in Central Africa. And in the West African clades here, we know that human strains found in southeastern Nigeria and although in many countries out of Africa, we know that their strains evolve approximately twice faster than other human strains in other locations in West Africa. So those of the clade 2A. And next slide, please. Next slide, please.
Oh.
Le slide 3.
Next. Okay. Oh. Okay. Oops, not. Okay. So and for the other clades, and recently a new lineage was discovered in South Kivu. As you can see here with the long branch, this clade evolved approximately twice faster than in other part of Central Africa. So how to interpret this wide variation in substitution rates? Currently, the interpretation is that the high rates of monkeypox evolution are the consequence of epidemic spreading, I mean human-to-human transmission, and with ApoB3 editing, whereas the lower rates of monkeypox evolution are the consequence of zoonotic spillover, I mean independent host jumps from animals to humans. But it's important to note that most of the monkeypox genomes currently sequenced are from human strains, and that there are just a few monkeypox genomes from wild animals. And I list here only published sequence. So the first sequence concerns the Thomas Rope squirrel, which was collected in Democratic Republic of the Congo. And the three other concerns probably secondary host. All of them are primates, and they concern the Suti mangabe in Côte d'Ivoire and 2 individuals of chimpanzee in Cameroon and Côte d'Ivoire. So, next slide, please. So this means that we need new data from mammalian hosts to better estimate the natural rates of monkeypox evolution in order to better understand human epidemics. Next slide, please. So to address this issue, we would like to detect the monkeypox in squirrels. Different species of squirrel, of course, which is considered to be the main reservoir, Funicerius and erythris, but also other related species of Funicerius. And oops, next slide, please. Sorry. Okay. The advantages of this museomic approach is that a large diversity of specimens, squirrel specimens, can be used for DNA extraction. And by this way, we expect to provide a more comprehensive phylogeography of Funicerius species. But if we are lucky, we can also find maybe monkeypox virus in their DNA extract. And as shown by Manon previously, according to a previous study, we can expect to find 9% of positive result for the monkeypox in this kind of samples. Next slide, please. But as you know, that specimen can be older than 100 years. So in those specimens, DNA is of low concentration, and the molecules could be very fragmented with respect to fresh samples. Next slide, please. But there are two methodological solutions to increase the concentration of monkeypox in our ancient DNA extract and to manage the fragmentation of the molecules. One solution is to use DNA hybridization captures with monkeypox probes. And by this way, we can increase the concentration of monkeypox in the extract. And the other solution is to perform next generation sequencing based on short reads rather than long reads in order to be able to sequence the smallest monkeypox fragments. Next slide, please. So just to conclude, we need to sequence monkeypox genomes from squirrels. First, to provide full evidence that Funicirus anerythris and maybe the other species of Funicirus are the main reservoir host of monkeypox. Next slide, please. But also, if we get a lot of success on museum specimen, we would be able to determine the geographical areas and seasons with higher monkeypox prevalence in squirrels. Next slide, please. And of course, with additional sequence, it will be able to further study the coevolution between monkeypox virus and its host reservoir in tropical African forests. And by this way, we will be able to test the hypothesis involving that the monkeypox virus diversification was possibly driven by Pleistocene climatic changes. Next slide, please. And at least with new sequences from squirrel, we can fully understand the history of monkeypox epidemics in Africa, and in particular to test the hypotheses involving higher rates of monkeypox evolution in West Africa, which could be linked to unidentified past epidemics. So thank you for your attention. Next slide, if possible. Okay. Okay.
Okay. Thank you so much. Thank you, Antoine. Thank you, Manon. Thank you, Alexander. It was a very, very stimulating presentation, information, full of information, full also full of research agenda. And of course, we have been, you have been explaining us your research strategy, which is very interesting because at some point we could have asked why to look at squirrel and so much in depth when probably the urgency is to fight only the pandemics. And you explained it very well what the identification of the reservoirs are providing to the scientific approach of the pandemics. I will not spend too much time because we are close to the temporally time limit, and therefore I ask perhaps Colin Kearney if he wants to ask his question, because Colin, you have two questions. May I give you the floor?
Yeah, thanks, Philippe, and thanks for the very valuable presentation. Yeah, I mean, my two questions are quite broad, I suppose. I have them in the chat, and you know, because we're seeing, we seem to be seeing more epidemics and in turn potentially pandemics arising. And is there a risk or that we will see more of these occurring in the future compared to the past because of different variables such as biodiversity and climate change, global travel, etc.
You hear me?
Philip, I think you need to be put on mute, Philip, if that's okay.
Okay. Yes, you are on mute.
So you're speaking about the possible emergence of other new viruses or other new epidemics due to all the different things that are going on, like climate change and globalization and travel and better notification and this kind of thing. This is absolutely evident. You know, if you look carefully over the last 50 years or 30 years, it has been a real increase. But the increase could be done to a better knowledge, to a better notification, but also to the fact that the world is a global village now, in few hours and in 1 or 2 days, you can transport, different person infected by viruses from one place in the world to another one, like it has been done for the COVID, for example, or for the MERS at the beginning. So it is sure that this will increase, and probably, we are waiting, I don't know, for a new coronavirus or something which can be really happening is a modification of flu, like avian flu, for example. For the moment, is really something which is quite hot. So generally speaking, this will very probably happen. So we need absolutely better preparedness, and also we need a rapid epidemiological survey, but the most important is not only to know which disease is going on. Of course, this is very important, but due to, for example, sequencing data in few days or few weeks, you know exactly what you are talking about. But, modification of the behavioral humans is very difficult, and this is really what is more difficult. For example, for COVID, very rapidly we know what was the COVID, but it was very difficult to stop the epidemic story. And this is more difficult, like HIV and many others. So,
Thank you.
regarding the...
Can I answer?
Yeah, sure, you can ask.
Also on deforestation, like the animals will not have, like a lot of mammals will have to like change environments. And so there will be more contacts between humans and animals, and this will lead to a lot more zoonosis.
Yeah, that's absolutely evident. Also, the increase of human population. This is really something which is really true. As more as you increase the number of contacts, you will increase the number of sporadic and pandemic disease. What makes a virus more severe than humans? For example, monkeypox versus chickenpox. So first, it's difficult to answer really this clearly, but if you look, for example, about monkeypox, the severity of monkeypox today is really linked not always to the virus itself, but the place where you are living. This is extremely important. For example, I told you that there was quite a lot of children dying of monkeypox currently and, the years and years before. It is mainly because there is no good antibiotics, there is no good availability of rehydration and these kind of things. And very probably, if the monkeypox epidemic was happening in Europe, for example, I'm speaking about children, there will be very little deaths or not deaths at all among children, despite the fact that we are discussing about hundreds of children for the last few weeks in Central Africa. So this is also a very important thing. I cannot really answer the question of the modification of viruses between monkeypox and chickenpox.
Yeah, but I understand. Yeah, I think it's very important to understand that it's the reactivity and the treatment of the actual virus is what distinguishes probably a lot of the mortality. It's how the prevention and the acting on the virus, where, as you mentioned, which brings a big difference in the mortality
in
between different
lethality
areas.
and case fatality, right?
Yeah.
Another
Yeah.
example, which is really very sad. You know, I told you that the very rapid diminution of the pandemic was due to behavioral modification, but also the use of vaccinia. Okay, the vaccination against monkeypox was quite a good vaccination, which quite efficient. And for the moment, despite the fact that in Central Africa it has been known for years and years, and despite the epidemic and despite the WHO alert, the vaccination in Central Africa, especially in DRC, just begin now. And it's quite difficult to get the vaccine, to get all the cold, chain and all the... It's very difficult. So which population to focus on? Here it was, mainly men were sexist men in great majority. When you speak about heterosexual transmission, this is very difficult to focus on, and you are not going to focus only on women or young women, on people living in mining activities and things like that. So this is really, all the logistic is extremely important, locally, to have a better administration of the virus, of the vaccination.
Okay, thank you.
Okay, thank you. I don't see further question. I would have a question on my side, but I think time is now going out. I propose, in order to remain inside the time limit we have fixed, and which help our participants to plan their own participation, I will close the webinar. But before that, I would like to thank so much Antoine, Manon, and Alexandre for this very highly valuable presentation, in-depth presentation, convincing presentation, and stimulating presentation. Thank you so much to all of you, and thank you for this valuable partnership we have with the Institut Pasteur, and of course behind it even the Muséum d'Histoire Naturelle. Thank you so much.
Thank you.
Okay, thank you very much.
Thank you.
Thank you.
Bye.
Goodbye.
Thank you.
Bye-bye.
Bye-bye.
Il n'y a plus que...
Il y a plus qu'à